Western blot gives us information about the abundance and the size of proteins in a complex mixture. We're going to use electrophoresis again to separate these macromolecules by size, but proteins are more complicated than nucleic acids because they have different charges associated with each of the potential side chains in the primary sequence of the protein. So we can't just assume that the charge to mass ratio will scale linearly with the size of the protein like we can with nucleic acids. One protein may be positively charged and another may be overall negatively charged. Since electrophoresis separates molecules by charge to mass ratio, the first thing we have to do to get a reliable size from a protein is to make the charge to mass ratio the same. So in a Western blot, we're going to use denaturing electrophoresis. The first thing we're going to do is take the proteins and we're going to mix them with a detergent. So any protein that is mixed with detergent is going to have the hydrophobic core of the protein disrupted by the detergent molecules. They are going to have these hydrophobic bits, the little squiggly bits inside here, and they're going to also have, the detergent's also going to have a negatively charged head. So the detergent will disrupt the protein molecule structure, and the hydrophobic bits will bind to the hydrophobic parts of the protein, and on the outside of this combination now we'll have these negatively charged heads from the detergent. A common detergent used in the process is sodium dodecyl sulfate, or SDS. So we call these SDS gels or denaturing electrophoretic gels. We usually separate proteins using a matrix called polyacrylamide. So the technique is usually called SDS polyacrylamide gel electrophoresis, or SDS PAGE, as it says over here. Okay, so once we've taken all the proteins in a complicated mixture and we've mixed them with detergent, now all of them have the same charge to mass ratio because they've all bound approximately the same amount of sodium dodecyl sulfate, and so they all have about the same number of charges on the outside because the complex of the detergent with the protein is more or less a uniform mixture. Okay, so now we've got all of our proteins denatured. Their shapes don't matter anymore. They're more or less unfolded, and they have about the same charge to mass ratio. So now we can apply an electrophoretic current and separate them all by charge to mass ratio. So here's a polyacrylamide gel slice over here. Again, these aren't usually run horizontally like we did with the agarose gels for DNA. These are usually run vertically, so they'd be sitting upright with the top up here and the bottom down here. So we just fill these up by gravity, put them in a solution, and let them flow into a well that has walls to it so it'll hold the complicated mixture. Now we apply a current, again positive at the bottom because we've got negatively charged molecules overall over here because of the detergent mixed with our proteins. So these guys, when we apply a current, are going to run through the polyacrylamide gel, and they're going to separate by mass because they all have the same charge to mass ratio, so they're all migrating with the same force through the gel. But small things can get through more easily than large things. So we're going to get small stuff down here at the bottom of the gel migrating faster, and large things up here at the top of the gel migrating slower. After we've separated the proteins by size, we can then take this gel, and once again we'll blot it to a membrane. So there's a membrane here, and now the pattern that we got with the SDS polyacrylamide gel electrophoresis is retained. That is, the big things are up here and the small things are down here. But now we've separated them by size and immobilized them on this membrane. Now I can take that membrane and soak it with some antibody that will recognize the protein. Now most antibodies will recognize a fairly small epitope, but these proteins have been denatured, so sometimes the antibody works and sometimes it doesn't for a Western blot. But we have to find an antibody that can recognize the protein we're interested in specifically, and will only bind to it after we've transferred it to the membrane. So the antibody is not going to bind to that protein because it doesn't recognize it. It's epitopes, it doesn't bind to that, but it will bind to the one against which it was first prepared. So we've done something to label this antibody now. In this case, I've put little gold dots on it, but that could be a fluorescent dye, or it could be an enzyme that's going to make a colored reagent like we did with immunohistochemistry. We can use the same tricks to detect the antibody. We can even come in with a second antibody that has a label on it that recognizes the first antibody. In any case, we have some way to detect the antibody, and now when we develop this gel, we won't get any signal anywhere except where the antibody recognized the target protein. So this is a Western blot. The amount of signal that we get is going to be proportional to the amount of protein that was in the complicated mixture. So we can determine both how big the protein was, whether it had additions or deletions or was proteolyzed or whatever had happened to it. So that'll be revealed by the size, how far it migrated into the gel. And we can also tell the abundance by how much signal we get. Less signal for a less abundant protein, more signal for a more abundant protein. So that's the kind of information we can get from a Western blot. As a practical example, we can detect, say, the amount of p53 protein in a sample. So here we've taken a time course after we have treated some cells with a DNA damaging agent, in this case X-rays, at an early time point, 0 hours before we've hit them with the X-rays. P53 is not very abundant, so our Western blot detects a very small amount of P53 protein present. After we have hit the cells with X-rays for 8 or 24 hours, there's still DNA damage in the cells, so the P53 level has been stabilized. We're going to get an increase in P53. So we can see that at this same size, we're getting more protein. So we've detected both the size and the abundance of the P53. At the same time, you can see that it takes 24 hours for the downstream target of P53 as a transcription factor to activate the P21 cyclin-dependent kinase inhibitor. In response to the DNA damage, P53 was stabilized here at 8 hours. It went off, bound to the promoters of various genes like P21 and caused them to be transcribed, but of course there's a delay because that process takes a while. So P21 accumulates later than P53 does. This down here at the bottom is an actin loading control. We're not interested in how much actin there is in the cells. We just want to make sure we've loaded about the same amount of protein, representing the same number of cells in each lane. So we've done another Western blot here, the same blot, but now we've probed with an antibody against actin, not because it's interesting to us, but because we want to make sure that the reason we're seeing more protein in the other lanes with time isn't because we loaded more protein. It's just because they were actually accumulating relative to the average cellular protein.